Hello everyone, this is Alexander Kuklin. Next is the news with Christina Carmichael. Christina?
Thank you, Alexander. Welcome to another innovative healthcare news segment brought to you from the Biopharma Institute. Today the news stories are: a test to predict acute myeloid leukemia was cleared by the FDA. Another story, a cutoff level for a biomarker used in a clinical trial with pancreatic cancer patients has been established by Clavis Pharma. Story 3, the FDA granted 510(k) clearance to Ventana Medical Systems for Companion Algorithm HER2 4B5 for image analysis applications. Abbott received clearance from the U.S. Food and Drug Administration for a new vitro diagnostic test to aid in determining the prognosis of patients with acute myeloid leukemia, AML. The VISIS EGR1 FISH probe kit, the third Abbott FISH assay approved or cleared in the past 2 months by the FDA for oncology applications, detects a chromosomal deletion in bone marrow that is usually associated with an unfavorable prognosis for AML patients. Abbott's VISIS EGR1 FISH probe kit can identify which AML patients have the chromosomal abnormality upon diagnosis and provides physicians with another clinically validated tool to assess a patient's overall prognosis, says Stafford O'Kelly, head of Abbott's molecular diagnostics business. Clavis Pharma announced the establishment of the cutoff level for defining HENT1 low versus high expression that will be used to assess the results from the ongoing LEAP clinical trial with their drug CP-4126 in patients with pancreatic cancer. HENT1, human equilibrative nucleoside transporter 1, is a protein expressed on the surface of cancer cells and has been shown to be important for the uptake and efficacy of gemcitabine. Low HENT1 levels have previously been correlated to poor clinical outcome with gemcitabine, the standard treatment for many solid tumors. This correlation has also been seen in the studies used to determine the HENT1 cutoff for the LEAP trial. The study of HENT1 expression in patients with pancreatic cancer was initiated using an analytically validated immunohistochemistry assay that measures HENT1 expression in tumor tissues. The study's objective was to define the cutoff for low and high HENT1 levels determined from pancreatic tissue samples obtained from gemcitabine or 5-FU treated patients with known disease outcome in a previously conducted randomized controlled clinical trial. Key findings from the study include maximum statistical separation of patient survival between the low and high HENT1 groups was found using a method of tissue analysis combining the area of HENT1 staining with the intensity of staining. The HENT1 cutoff level determined a highly statistical significant difference in overall survival between low and high HENT1 expression in gemcitabine treated patients. Importantly, there was no difference in overall survival between low and high HENT1 expression in 5-FU treated patients. While approximately 2/3 of tissue samples tested were measured as low HENT1 based on this newly established cutoff, no assurance can be given that this will be the same percentage in the LEAP trial. Ventana Medical Systems Inc., Ventana, a member of the Roche Group, received 510(k) clearance from the U.S. Food and Drug Administration, FDA, for the Ventana Companion Algorithm HER2 4B5 for the image analysis applications with associated Virtuoso software and iScan Creo Gold scanner. Ventana is now the only company with a complete workflow solution for determining HER2 4B5 expression in breast cancer patients. This application assists the pathologist in the detection and semi-quantitative measurement of HER2 4B5 protein in formalin-fixed paraffin-embedded normal and neoplastic tissue. While the pathologist is still the ultimate authority in scoring HER2 4x5 stains, the image analysis application helps ensure consistency and objectivity in interpretation. Thank you for watching. This is Christina Carmichael with Innovative Healthcare, a service from the Biopharma Institute. Back to you, Alexander.
Thank you, Christina. And once again, thank you for joining PMTV tonight. Come back for new programs. Good night.